Okay, so it's 3:02. Welcome everyone to our fourth online scientific symposium. For this session, we're going to be talking about data gaps in toxicology, whether the result of selecting publishing practices or a simple lack of available studies, and how to overcome this challenge. For those of you who don't know me, my name is Paul Whaley, and I'm a research fellow at the Evidence-Based Toxicology Collaboration, and I'm your moderator for today, backed up by my fellow colleague at EBTC, Dr. Sebastian Hoffman. In case you're not familiar with EBTC as an organisation, we're in the business of promoting high-quality, high-impact research in toxicology and environmental health, where we work on raising research standards in toxicology, making sense of evidence using systematic reviews, evidence maps, and other evidence synthesis products, and improving access to research through fair data and open science, and advocating evidence-based decision-making, including integrating evidence into policy-making. We've been quite busy over the last 2 or 3 years, developing systematic methods for adverse outcome pathways, the Costa recommendations for good practice in toxicology systematic reviews. We've been working with editors to improve publishing standards. We've been evaluating non-animal test methods for drug-induced liver injury. And then coming soon, our next paper, we're updating an understanding of Bradford Hill's concept of biological plausibility for environmental health systematic reviews. We're funded mainly by the Centre for Alternatives to Animal Testing at Johns Hopkins University. We have in-kind contributions towards projects from US FDA, Tox Strategies, Norwegian Institute of Public Health, and Symposium, and we're very appreciative of that. If you want to join the collaboration, we operate on a Cochrane-like model, running collaborative projects through thematic working groups. We're going to be launching a new network structure this summer. To join, I recommend you subscribe to our newsletter, or you can email Katya Sayun, our Executive Director, directly. So, we have 3 speakers today talking about overcoming data availability obstacles in the way of evidence-based toxicology. Our first speaker is Katya Sayun, who is our Executive Director, and is presenting EBTC's new analysis of Tox21 data as a case study in the challenges of evaluating drug safety when only some of the relevant data is publicly accessible. Then we'll be hearing from Jan Turner of the Safer Medicines Trust, who is presenting on gaps in preclinical and clinical data around drug discovery and the need for transparency in the evidence base that regulators are using in making drug approval decisions. And then finally, Professor Thomas Hartung, Director of the Centre for Alternatives to Animal Testing, will present his work on using artificial intelligence and machine learning methods to fill in the gaps when studies related to chemical toxicity are simply not being conducted. I hope you enjoy the presentations. Each is 20 minutes long, and there'll be a panel discussion for 20 minutes after the talks. Please drop your questions for the presenters in the Zoom Q&A window. For the speakers, I'll let you know when you have 5 minutes and 2 minutes left in your presentation time. The call is being recorded and it will be uploaded to the EBTC YouTube channel in the next few days. So Katya, I'm just going to hand it over to you.
Hello, everyone, and welcome to our the EBTC's fourth webinar. So it's my pleasure to present the data from one of our systematic reviews that started a few years ago. And the main conclusion from that, just to start where I will land my presentation, is that there are a lot of data gaps in the toxicology evidence, right? So the Tox21 project was a collaboration between EBTC. It was EBTC working group that's co-run collaboratively with Norwegian Institute of Public Health. And it was co-managed by Hubert Dyrven from the Norwegian Institute of Public Health. And a lot of work on this project was also done by Gunn Wist, his colleague, as well as the other 2 members of the working group who are mentioned on the slide, Shri Bandakavi, who was responsible for the ToxCast data analysis, and Jyotsna Mehta, who did the human evidence events analysis. So the project has started with the question: why do the various drugs, sometimes from the same class, induce liver injury while others do not, right? So so let's look at the drug discovery and development process, which is, as we all know, is very lengthy and expensive. And if you start from the very beginning, on the left, it starts with target validation, which typically is not done in a pharmaceutical company. It's usually done there's a lot of scientific work that happens before the drug discovery program starts, including the target validation, whether the target is druggable and things like that, and whether it's related to a particular disease. So once the target is identified, then the drug discovery program can start. And this results in a small molecule space from many candidates in screening, typically high throughput screening, and then formal preclinical development that involves one rodent and one non-rodent species, which is usually a dog and a monkey, for safety evaluation prior to clinical trials, which start with safety in healthy human volunteers in phase 1, and then phase 2 and phase 3 with increasing number of patients. Then finally, there is an IND submission and FDA review, which can happen, which takes anywhere from 6 months to 2 years. As we all know, with COVID pandemic, this process has been significantly accelerated, but whether it's going to stay, keep some of those features and urgency remains to be seen. But typically, the drug discovery process takes on average about 12 to 15 years. So what happens in this whole process is then there is a so-called phase 4, which is frequently called like that. It's a real-world evidence or post-marketing surveillance for safety. And in this process, what happens is that sometimes the signal for safety is being detected, and then the data is reviewed by FDA quite rapidly to address any safety concerns, right? So in this study, we investigated the case of 2 drugs, troglitazone, that has been approved to go on the market. And within really the first 2 quarters of being released on the market, the signal for safety has been detected. So the drug has been withdrawn from the market within less than a year from being approved. The other drug in the same class that has been released a year later still remains on the market. It has other issues, but no severe drug-induced liver injury, which has been detected for troglitazone. So this, of course, is not something that anybody wants. It results in human suffering and loss of life and disability, and also has tremendous cost for pharmaceutical companies. So troglitazone disaster cost Pfizer close to $10 billion in lawsuits and loss of revenue. So both of these drugs are from the same class. They're both anti-diabetic drugs, and there was a good rationale for inhibiting a particular enzyme, the thiazolidinoldeones, sorry, from the same chemical class, and they they inhibit PPAR gamma, which is supposed to reduce the glucose, right? So, and what happened, the FDA data that resulted in removal of troglitazone from the market is shown here in this graph, where you see the increase in the reports of severe drug-induced liver injury in troglitazone. So this is the design of the study. So when we set out to do this study, we wanted to conduct an evidence-based evaluation of all available evidence of those 2 drugs and see if there is any particular test method that would have detected the difference between the 2 drugs. So to do so, we conducted a systematic literature review of in vivo studies, both animal and human. We took the largest publicly available in vitro database, US EPA ToxCast, and extracted data on those two drugs. And finally, we looked at the human adverse events reported in the largest WHO database called VGBase. So in essence, we had preclinical and clinical in vivo data, in vitro activity for these two drugs, and the human post-marketing data. I'm not going to talk about the human post-marketing data because even though we conducted our own analysis here, which is reported in the paper, but this basically confirmed what we already knew, that troglitazone had significant drug-induced liver injury, whereas rosiglitazone didn't. In the systematic literature review in the in vivo evidence stream, we have conducted the protocol-driven systematic review of the literature. So we reviewed over 9,000 studies and went through the two phases of title and abstract and full text screening. We identified 690 papers that were eligible for screening, and then we excluded 649 of them for these various reasons. Majority of them were because there was no primary data. And finally, we ended up with the 42 in vivo studies. And what we found in this exercise is that there was no huge difference. There was no clear signal in the in vivo evidence, both reported animal and human, that troglitazone would have gone on to cause those drug-induced liver injury. It certainly confirms the FDA decision to get the drug approved. So the evidence stream. So what we found, the limitations of the published literature. So there is a significant publication bias, which is the topic of this seminar. The regulatory preclinical and clinical studies tend not to be published. They are considered proprietary sponsor information. So we need to have incentives for industry and regulatory agencies to make the data public. We also noted the publication quality. There's a frequently lack of detailed protocols, lack of individual animal data, insufficient details in the methods, and also selective reporting. In toxicology in particular, this is a big issue because nobody wants to publish the data on chemical being safe. If you give it to the rats and the rats are happy, this is not particularly interesting. If you find some pathology, that's when the data becomes publishable. So a lot of that data is actually not reported. The difficulty in the screening of the literature is lack of structured abstracts made it particularly difficult and lengthy process to select the relevant literature. So now moving on to the US EPA ToxCast in vitro data. So you see the workflow of how this was done. This was performed on the left, and this is the work of Sri Bandakavi, as I mentioned. So we did not pre-select the tests that would be relevant to liver. We took the entirety of ToxCast database, and the way the data is publicly presented is there's AC50s, 50% inhibitory concentration of various enzymes. And we looked at the tests that were done, were performed for both drugs, for rosiglitazone and troglitazone. So there were 437 tests that identified positives, right? So the number of positive tests for troglitazone was twice as large as for rosiglitazone. So immediately you see that there is much more off-target activity for troglitazone, which has been withdrawn from the market, compared to rosiglitazone, which is still on the market, right? And if you remove the tests that you could consider potentially promiscuous, which detected a signal in both drugs, then the difference is even larger. So you have 78 positive for troglitazone only, as opposed to only 10 positive tests for rosiglitazone. So So as a result, so the in vitro tests provide insight into biological pathways that are activated by both drugs, right? So they provide a clear differentiation between troglitazone and rosiglitazone. So there are also limitations in ToxCast data set. There is missing data, so not all compounds were tested in each assay. The metabolism at the time that we looked at that data was not present. The tests that were selected in the database that were chosen some 10, 15 years ago and not always related to the prevalent mechanism. And also there are some other models now that actually include metabolism. There's 3D microphysiological systems and whatnot that are more, that recapitulate more the human biology. So what we've learned in this exercise, the limitations of the systematic literature review, of course, is the publication bias, the lack of published regulatory animal toxicology studies is particularly needed in order to fully evaluate the toxicology of the drugs and other chemicals, and lack of published human randomized controlled trials is also very obvious. There were only a few very small randomized controlled trials in both drugs that we found that are published, and it is obvious that the drugs were approved based on large number of patients and large data sets, which are not available publicly. And also, of course, the reporting deficiencies and positive result bias. So in the ToxCast, as I mentioned, there are significant limitations as well. There is also varied AC50 calculations, not relevant to all endpoints that we found that were used, and the missing data. There's certainly a need for having a database that contains more assays and better reported data. Finally, in the VG base, so that's the limitation is that the main limitation is because that's not something that you can do when you're developing the drug. This is something that's already happening or has happened, but it does provide the evidence-based information about the factual information about the drugs, and it's a human evidence. So that's a good sort of denominator to compare your preclinical and mechanistic data. So on existing drugs. So what we've also found is that there were a lot of developments in that facilitate development of conducting the systematic literature assessment, systematic literature reviews, such as AI-powered software, which is Distiller, Scion Active Screener, Scion and SysRev that we used in this study. The machine learning and AI certainly helps in screening the literature faster and screening out the articles that are completely off topic, which always happen in a systematic literature search. So, but the biggest gap that we found is really in the systematic literature review, especially, is that regulatory agencies really need to find ways to make the data public, and that's a big gap.
So, Kat, you have 5 minutes.
Good. We have 5 minutes for questions because I'm done. Essentially, where do we operate here? So of course we have, this is a known risk assessment matrix, right? So we know we have known knowns, which are facts, right? So that's something that we can easily assess. We also have known unknowns, which are the questions that we can pose, right? But this could be internal company data and regulatory approval data, which is, we know that it's available, we just don't have access to it. Then we have unknown unknowns, so we know that we don't know all the biology, and there are things that we can certainly learn. So that's something that's where active research is going on. And finally, we have unknown knowns, and that's where our intuition and prediction and more prediction tools and AI are becoming relevant, which is the topic of Thomas Hartung's talk. So I'll end here, and we can have a few minutes for questions if you want, Paul. Thank you.
So thank you, Katya. I don't see any questions in the Q&A just yet. I just remind participants that if they want to ask questions, that's the place to do it. Katya, so knowing that so much data is not publicly available in toxicology, right, bearing in mind how long it took to do this study, does it actually, in your opinion, make sense even to embark on systematic reviews on chemicals where so much data ends up being unavailable?
Yeah, I think the systematic review in toxicology is roughly where the clinical research systematic reviews were about 25, 30 years ago, right? So the situation was very similar. So there were not many randomized clinical trials to do systematic reviews on. However, what it does, what this method allows is identifying the gaps and identifying the issues with the design of primary studies. So, and that's, so it's not a futile exercise. In this exercise, we have learned a whole lot about design of the primary studies and about the data gaps that we have identified. So that allows the researchers to take note of this. And as more systematic reviews being published, this is going to become drive the research to improve the quality of research and also to get more predictive models in place.
Brilliant, thank you. Right, so we're coming to the end of the time for this presentation, so I think we've got Jan Turner on next. So what I will do is, oh, we have a question as well. So what we'll do is we'll just save that question for the panel discussion. Jan, if you'd like to share your screen, then you should be able to start with your presentation.
Great, thank you, Paul. Can you hear me okay?
Can hear you fine.
Great. Okay, and you can see my slides.
Yes, we can see your slides as well. So I'll let you know when you've got 5 minutes and 2 minutes.
Right. Great, thanks, Paul. Okay, so thanks, Paul, thanks, Katya, and thank you to EBTC for giving me the opportunity to speak at today's meeting. And to follow on from Katya's talk, today I'm going to talk about gaps in preclinical and clinical data and why there is a need for transparency in the regulatory evidence base. Before I do, I just wanted to tell you a bit about the charity I work for, and this is the Safer Medicines Trust, which is a independent UK-based patient safety charity. And our aim really is to make medicines safer through the greater use of more human relevant science. We're also founding members of the Alliance for Human Relevant Science and recently produced a white paper calling for the acceleration of growth in human relevant science in the UK. And we're also actively engaged with politicians on an all-party parliamentary group for human relevant science. So my talk today will cover what the problem is around data transparency and the implications this has for both development of new and safer medicines. I will share some examples of these issues in both preclinical and clinical areas, and also touch on some initiatives in various regulatory sectors that are embracing regulatory sectors embracing this to increase transparency. So to start, what is the problem? There are a number of issues relating to lack of data sharing and transparency, including, as Katya has already mentioned, the non-publication of, for example, negative data. The quality of the data itself may be lacking and questionable. There is a lack of standards around reporting data, especially in the preclinical space, and there may also be bias in reporting from drug sponsors, including selective removal of data. So improved transparency in data is needed for a number of reasons, including reducing the number of repeated experiments carried out in order to reduce costs and resource use, to increase the knowledge gain that can be achieved, and to improve efficiency of trials and clinical outcomes. And a specific question in terms of the preclinical space is where we work is, are regulators actually sitting on data from sponsors and developers, which if made available could potentially help the above points. So I want to move on to the impact of poor reporting of preclinical results. And in the next couple of slides, I'll discuss different areas mentioned previously and what the outcome of each of these was. So the first publication is from one of our research consultants at Safer Medicines Trust, Pandora Pound. And this was a retrospective harm-benefit analysis of preclinical animal research for 6 treatment interventions. The animal studies were assessed against the benefits to humans, and for all of the studies it was found that they were of poor quality, and less than 7% of the studies were actually permissible according to the Bateson cube, which is a method of determining the cost versus benefit of animal research. The findings also indicated that there was a need to review the current regulatory processes current regulations and processes for assessing preclinical animal studies. But ultimately, this work highlighted that the benefits that may be achieved should be based on more scientifically rigorous research. The second example is a case of selective reporting, and this was the development of a tuberculosis vaccine booster by Oxford University. And this was investigated by the British Medical Journal in 2018. Here, results from non-human primate animal studies from Porton Down, UK, were selectively reported at various stages in the development of the vaccine, both to gain more funding and also for the approval of human trials. Unsupportive data was excluded, and this resulted in major funders of TB research actually rethinking their priorities and stating that this had actually slowed progress in the entire field. And here you can see in more detail where significant data were admitted. And in conclusion, the British Medical Journal investigation asked whether the parents of babies that were included in this in subsequent clinical trials, this actually went to phase 2 trials, were actually misled. This third example, I won't dwell on Katia, her presentation, but it's clear that, that in this recent publication, there were a number of areas of poor reporting which limited the possible analysis of data and conclusions reached. And I would agree wholeheartedly that we need things like tips for industry and regulatory agencies to publish this data. Finally, in this section, publication where 210 archived animal study protocols from 2 major German university
Jan, we're just getting a couple of issues with your audio. You might want to disable your webcam just to see if we get a bit more bandwidth that way.
Let me do that, hang on. Oh, come on. Can't do it.
Jen, I can stop your video. You can just go ahead.
Could you do that? Yeah,
yes, I can do that. Yes, I'll do that.
Apologies. Thank you. Okay, can you hear me okay now?
A little better, and if you want to put your presentation back, thank you.
Okay, great. Yes. In this example, this is an example of publication, and here 210 archived animal German university medical centres were tracked in terms of their results publication, and overall around 33% of all animal studies were not published. This study also confirmed that the practice is relatively common, but standards and policies in place to prevent this. It was also suggested that pre-registration are helpful. In terms of pre-registration, this has also been suggested by others, and in the Netherlands there is a Preclinical Trials.eu web-based platform which is available database animal studies, which is funded.
Jan, I'm afraid we're really struggling to hear you at the moment, so I think what we might do is, can you try dialing in using the phone number that you got with your participant email?
Yeah,
sorry everybody about this, we're just having a couple of technical issues.
no, it's probably my end, I'm sorry. I can't actually find a number.
I will resend it to you. It should have been in an email that came from Zoom this morning, Jen.
For some reason, I wasn't getting emails, so each time I've gone in, I've had to re-register. So I'm sorry about that.
Yeah, let me try to resend it to you.
Thank you. Apologies for this.
That's okay. So while we're just getting this sorted out, what I can do is I can put a question from one of our participants to Katya and probably to Thomas as well. I think he might have an opinion on this. So, so SEND is a new standard for exchange of non-clinical data. Katya or Thomas, I don't know if you have a view on how that may change the problem with access to preclinical data.
Yeah, so I can start. I haven't actually used the SEND personally, but as I understand, this is an electronic system for data submission of IND documents to FDA. So it certainly is a step towards making easier for the FDA to make the data available. So, but there's still, as far as I know, there's no requirement to make that data publicly available.
From my point of view, the problem is the lawyers. The FDA is not the owner of the data, so they cannot publish them except there's a legal requirement for publishing them, which means it is about the companies. Whether they are willing to publish, and while the scientists very often agree that these data are helpful, that they're pre-competitive, lawyers very often say, why should we if we don't have to? We are giving away on what we are working. We are opening up for possible liability cases because we could have seen something which we did not notice. So it is not about the standards on how to share it. Once we agree to share, then it's very much about standards of communicating them that they are actually usable. And a lot of the problem is really about the metadata, and these standards are extremely helpful because they clarify it's not only about sharing data, but it's also about what these data actually come from.
Yeah, and then it goes to into the data interoperability and the ontologies. So it's all becoming very, very complicated. But that's the baseline where things should start, because you need to have the common language of how you call things, and that's frequently also different in different fields and subfields.
I mean, basic science is really getting better. There's a general commitment to the FAIR principles of making data available and accessible and findable, and this means that, and this is something NIH requires, for example. But the 90% of the work in toxicology is roughly done in companies and in a regulatory context. And that's the stuff which would really be important to share. It also has the highest quality standards. And this is the one which we don't get.
Very good, thank you. Camilla, do we have Jan on the phone now?
I don't believe she's able to dial yet. Jan, can you still hear us?
I can hear you. I just don't have any details on dialing in. I'm
Yeah,
so sorry.
I did send you an email, so it should have been from Zoom specifically.
We do hear you now, so maybe you could try to share your slides again, and
yeah,
we can turn off our videos and just keep going.
Okay, I'm sorry about this. Yeah, there's some issues I think with Zoom for me, but okay, I'll continue then, shall I? And,
yeah, we'll see. We'll let you know if there are any other issues, and if necessary, we'll bump Thomas's presentation up, and then that'll give us 20 minutes to try and sort you out.
Yeah,
okay.
Jen, the other option also is I send you a link as for you to come as a participant, and I can always let you, you can dial in as a participant, and I can make you a panelist, and that way you will have the access to the audio.
I think we should try to pound through right now. Thanks. Yeah. Thanks, Camille.
Okay. Let me know if you can't hear me anyway. Okay, so I think I got to this slide here where we were talking about pre-registration of preclinical research. And I was mentioning that the Netherlands have a database, preclinicaltrials.eu, which they hope to expand out internationally, and also Germany, for example, have a similar registry that was set up in January 2019, which is available and actually is embedded in the infrastructure of the German federal government. So they claim that continuity and data security, as I think I heard you say, Thomas, is guaranteed, or is at least provided for sharing of that data. So this is not a new idea, and I think it's something that a previous FDA commissioner, Robert Califf, suggested in 2016, and he referenced the main clinical trials database, clinicaltrials.gov, as a model upon which preclinical registration could be based. He stated that there was, however, no compelling reason for people to actually publish their data, because if the results were favorable, they became trade secrets, and if they were unsuccessful, they were dropped, and no one really cared whether they were published or not. So having said that, a study of clinical trial reporting in 2019 also, in this case it was by the Science Journal, also found that many drug makers and academic institutions continually failed to actually comply with expectations of reporting, and only 17% of 216 trials investigated had the results submitted in the time period requested. And of 184 sponsor organizations that ran at least 5 trials, 30 of those never met an actual single deadline. And this really highlights the issue here of non-mandatory and the passive nature of reporting for clinical trials. So the goal really is to ensure that each trial contributes accurate and complete information, and eliminating so-called secret trials or biased reporting, which is misleading. Having said that, it was interesting in May this year that the International Coalition of Medicines Regulatory Authorities, ICMRA, and the World Health Organization released a joint statement on transparency and data integrity, calling for pharmaceuticals to do just this, to provide access to clinical data for all new medicines and vaccines, and that these should be published without reduction of confidential information for overriding reasons of public health. They stated that both positive and negative data should be made available, and it is owed to trial participants who contributed to these results. They acknowledge that regulators spend considerable resources on negotiating transparency with sponsors, but it's clear that many initiatives in place are still reliant on goodwill or actually lack appropriate resourcing. So I just want to move now on to some initiatives with various regulatory agencies, which hope to improve transparency of data reporting. But before I do that, I just wanted to mention SEND, and I know we've just been discussing that. I missed some of that conversation, but this does offer a standard by which pharma can report toxicity and safety pharmacology preclinical data. And as I think, again, Thomas mentioned, all IND and BLA submissions to the FDA from December 16 now have to be compliant with SEND. Other regions and agencies are in the process of investigating the potential of using these standards for sponsor submissions. However, Japan recently carried out an investigation into the quality of data in SEND domains and has recommended improvements in the quality of data sets themselves, as well as increased searchability before adoption takes place in Japan. So just turning on to some of the initiatives with regulatory agencies, the EPA is clearly committed to making evidence-based decisions on the best scientific data available. And to this end, they are looking to create flexible and sustainable processes for integrating publicly available data in order to prioritize chemicals for screening. Maureen Gwynn from the EPA described this proof of concept study at the CAT webinar back in May. What's good about this model is that it should be able to adapt to new scientific advances and incorporate new data in a modular fashion. The EPA is also committed, as we know, to reducing testing invertebrates, and an industry consortia has been formed to jointly conduct studies explicitly to prevent duplication of effort. NICETM, the NTP Interagency Centre for the Evaluation of Alternative Toxicological Methods, have their ICE database, Integrated Chemical Environment Database, which contains curated data and various tools from NICETM and its partners. And this allows free access to high quality data formatted for use in computational workflows.
Jan, you have 5 minutes, by the way.
Thank you. Data from both animals and non-animal tests are included in ICE, and chemicals are annotated for mode of action and phys-chem properties, etc. Katia previously mentioned the TOX21 database, a shared platform containing high-quality bioactivity data and assays, which includes over 70 screening campaigns and approximately 10,000 chemicals. More than 100 papers and 100 million data points have been generated from the TOX21 database, and similarly, the EU Tox Risk Knowledge Platform contains over 1,000 data sets and more than 140 peer-reviewed publications. Finally, the FDA Predictive Toxicology Roadmap that was released in 2017 describes leveraging research to identify data gaps and has identified a need to expand acceptable assays in regulatory submissions to include human-relevant in vitro and in silico methods. The formation of an alternative methods working group is going some way to achieving this, but many regulatory studies, and especially those that are not approved, are more often than not in the public domain. So there's still some way, I think, to go here. So in conclusion then, it's clear that we need transparency in the regulatory evidence base, and importantly, all data, whether negative or positive, should be available and accessible. This should help improve the quality of data available to researchers, but only with the implementation of standards in reporting to prevent bias and incentives to report can we really achieve progress, I believe. The benefits of doing this to researchers and the public alike are numerous, but ultimately this should improve the quality of data available and public trust and confidence in regulatory decisions. Finally, and perhaps most importantly, is that at the end of the day, trial participants have sacrificed their physical well-being and taken potential risks in order that better and safer medicines can be advanced. And we perhaps owe it to them and any patients waiting for new treatments to be more transparent and open. So that's all from me, and apologies for the gaps in sound.
Thank you, Jan. That was great. And well done on pressing through with that. That would have completely thrown me. So thank you for that and your patience with the technical issues. I think what we'll do is we'll just press straight on to Thomas's presentation now, and then we'll have a panel session afterwards. So Thomas, if you're there, if you have control of the screen, I think you should just be able to share and press on.
So, excellent. Yes, hello, this time from Italy, which is exciting after 1.5 years grounded in the US. Hello, everyone. Let me see that I get into the sharing mode here.
Hey, Thomas.
Yes.
I already can see that your audio and your video is a little bit delayed as well. Do you want to just go ahead and just use audio?
I am happy to stop the video flow. Good. Or can you stop it because I'm already in sharing mode?
Yes, I can.
Okay, good. Okay, so we are talking about data extraction, and what artificial intelligence can help us with this, and giving the examples from predictive toxicology to show the potential of these relatively new technologies. You know that I'm very much an advocate for replacing animal testing, directing the Center for Alternatives to Animal Testing. But these masks, which we actually printed, saying saving more than animals, is an important message for me. I'm a physician in the School of Public Health, and activities like the evidence-based toxicology collaboration, which is not alternative method with another name, but is trying to improve how we do risk assessments, is, I think, evidence for this ambition. I also would like to share a few conflicts of interest with you. I'm going to talk about some activities which were sponsored by Underwriters Laboratories UL, done with the spin-off company of one of my former PhD students, ToxTrack. And so please take everything I'm saying with a grain of salt. I'm trying to bring these things into application, and this requires me to get involved with some companies. AI has developed over the last 5 years into a major area of my work and our center's work. Among others, I'm now the field chief editor of Frontiers in Artificial Intelligence, a journal which just has been accepted in PubMed. So we are really doing a lot in the area, and I would like to share some of the learning experiences I had myself in this field most recently. So what's the problem? From my point of view, the problem is that we have one approach in toxicology which is dominant, which is get us one animal experiment, typically in rats, and this tool has to fit all of our problems. And we're using typically a sledgehammer approach, which is maximum tolerated doses into young animals, then we see what's happening. This is not an approach which reflects what our humans, patients, customers are exposed to. It is not really what we are interested in. So necessarily, science has evolved. And we see now that toxicology is flooded with information sources. We have more and more human studies, epidemiological, clinical studies. We see studies in lower animals, not only in the mammals. We see in vitro, in silicon, and chemical tools. And this brings us to one key problem. This key problem is data integration. Evidence-based medicine was relatively simple because they had to combine, at least in the beginning, clinical trials. We have to deal with in vitro, in silico, in vivo studies, and epidemiological studies to come to conclusions. And this challenge of data integration is something which is just the same whether we do a systematic review, where we want to bring in all the existing information, whether we do a risk assessment, which is also condensing all the different sources of information, or whether we are designing integrated testing strategies where we say, okay, we want that the different pieces of information help us to come to an overall result. So this is intellectually just the same. The difference is only in some cases we talk about data we have, and in other cases data we want to generate. But it's about the quality of the sources, how to weigh them, and how to combine them. That's the challenge. And what I would like to show you today is that there's a new kid on the block, which is artificial intelligence, which does exactly this. which is integrating tons of information and finding the way how to weigh it and how to pick the pieces which are really telling. So the question posed here is AI a different way of evidence integration? I would like to make this case in my presentation. The next thing, AI is really a new kid on the block. This is an article Sebastian Hoffmann and myself wrote in 2009 about computer-aided toxicology, so the in silico tools. And one is missing, which is AI. We didn't even think of mentioning AI because AI was not a major player in 2009 in toxicology. And the reason for this is that only since 2012, roughly, we have the high efficient algorithms of deep learning, which are at the moment taking over essentially every area of life, not only the life sciences. From self-driving cars to Google's algorithm to find you the best information, it's all driven by the new AI. So 2009, it was an omission, which is understandable. So AI, what is AI? In the most simplest definition I can make is it is making big sense of big data. Nothing you can do on an Excel sheet of data is really AI worthy. This is not big data. Big data is actually defined by at least 3 V's, possibly 5. The first one is volume. It really needs to be big. And big means terabytes. Big means a lot of connections between these data points. This is the second point. It is about variety. It is not just having the same piece of information, let's say on many chemicals. It is about a lot of different information. This can be structured, unstructured information, complete, incomplete. There's a lot in the bucket we work with when we're using AI. The third V is for velocity. It is about the accumulation of this information, which is taking place so fast that we are continuously expanding universe. You have to imagine of all of the information, all the data in the world, about 94% were produced in the last 5 years. Imagine 94% of all of the information which we have stored in the world was produced in the last 5 years. And this is only accelerating. And then there is additional things, which is value and veracity, so the incompleteness, the uncertainty of this data, which is often added as the 5 V's of big data, but the important ones are here really the message of complex data, which are permanently expanding. And there's a lot of tools which are at the moment delivering big data to toxicology. If you go through this picture here, the legacy data of the past, which are increasingly organized in databases, is only one of them. Whether it's PubChem or the integrated chemical environment of Nicetum, whether it is the EPA's dashboard or ToxRef database and whatever, we are getting more and more publicly shared databases available to work with. There's also literature. If you take PubMed as the most important biomedical database, they are getting 2.5 million scientific articles per year. And 80% of them describe chemical effects. 80%, 2 million articles in PubMed alone, describing chemicals in biological systems. And we have tons of gray data in the internet. We found 600,000 chemical safety data sheets, for example. There's sensor technologies which allow us to monitor our systems in real time. There's robotized testing like Tox21 and ToxCast. There are the omics technologies of genomics, transcriptomics, microanomics, or metabolomics. And last but not least, a lot is about pictures. It's about pathology done by high content imaging. So a lot of methodologies here which are feeding or providing us with big data which are suitable for AI approaches. And AI comes in different flavors. I will give you a few examples of the things AI is doing, what it means for toxicology. The first ones were really the expert systems. This is not a machine learning approach like most of the AI technologies nowadays are. Expert systems were really rule-based systems where we tried to put into formal rules what the experts were saying. And actually the very first paper I approached in toxicology I found was this paper by Rosenkranz from Case Western. He trained in 1985 on only 61 chemicals and applied it to one other chemical. He used the type of AI approach and an expert system. But already a few years later, this system trained on some 3,000 chemicals from the National Toxicology Program delivered some 98% sensitivity and 100% specificity in identifying carcinogens. So there was enormously fast progress out of this approach. And side note, our center actually did finance him from 1994 for some work on developmental toxicity. So this was the early work on applying AI type of tools. A lot is done on the vision side, image recognition, machine vision, but especially the interpretation of pathological slides of histopathologies and others. I have to refer you to a workshop report we did earlier on where high content imaging is standing. This is increasingly helped by AI technologies to support the pathologists and scientists to do the work. The next area of artificial intelligence is actually speech and text recognition. And this is what is often referred to as natural language processing. But it's also about mining literature. This is a scheme here, which is showing you how systematic reviews can nowadays be done. So the literature research you do, like Katja was showing you, can deliver you thousands of papers. And it's an incredible effort to read only the abstracts and sort what is in, what is out. Machine learning can help us with this because it helps us to eliminate the non-relevant ones and prioritize the more relevant papers after learning what we're interested in. It can help then with data extraction, help with study quality evaluation, and the analysis and visualization of what we really have in our bucket. And an example for this would be, for example, sysref.com, a program which Tom Lichtefeld, my former PhD student's company ToxTrack, has created. And this is interestingly free. So go to sysref.com and check it out. More than 3,500 systematic reviews have been done on this platform. And it is providing you with possibilities to auto-extract, for example, from PubMed or other sources, to auto-annotate papers after learning circles, and then to cluster analyze papers and learning from what you would include and exclude, what is in the bulk of articles really worth looking for. I think this is a very interesting way of shortcutting systematic review processes. Very closely related to speech aspects is natural language processing. So not only speaking and understanding text, but really to process language. And an example here from our own work was when Tom Lichtefeld, again, in his PhD, made by natural language processing the ECHA registration database machine readable. Because the big problem is that these wonderful open access files, which are providing us with the information, the core information of the registration process, that they all come in unstructured texts and a lot of PDF documents. And you have to teach the computer first what it is reading before you can really extract it. And in December 2014, we found already 10,000 registration dossiers, which included more than 800,000 toxicological studies. This was billions of dollars of testing information. And these 10,000 chemicals, we extracted with natural language processing. That's Tom, by the way, just to give him some credits here. This was instantly taken up by the press. Here, there's an article here in Science. There's an article in Nature. We reported on this treasure trove of a database. And quite importantly, while ECHA was originally a bit irritated, they very quickly clarified that this is in the best interest of animal safety, chemical safety and animal welfare. And they confirmed that this type of extracted data can be used for predicting chemical properties. They cannot be used for one-on-one predictions of or registration process, but you can extract them like for a QSR or similar tools. And this REACH database is until today a critical resource in the field because it is very different to all other toxicological databases full of negatives. As Katja rightly said, the literature is full of positive findings because it's boring to tell people I didn't see something on a chemical. REACH is full of this because these are chemicals which went to consumers and did choose the ones which had no toxicological effect. The next element, which is what we nowadays understand as AI, mainly is machine learning. And machine learning comes in, again, many different flavors. We are using it, for example, mainly in Alex Mertens' work, to make sense of omics data. So we're trying to use machine learning to understand what do transcriptomics and metabolomics data say. So this is an example where she translated weighted gene correlation network analysis to metabolomics. There was other work where she studied endocrine disruption from large databases on bisphenol A, or where she studied cancer gene expression, asking, are we studying the right genes? It's all mining these large data sets with these type of machine learning tools. More recent work, more recent work makes sense of omics data, trying to use very similar approaches of mining larger data sets. We converted in work of Tom, his database into a predictive tool. And this is the work we did with Underwriters Laboratories. So we expanded the REACH database, which we had with other public sources, to achieve a database of some 10 million structures. We created with the Amazon cloud server of 180 cores and 2 days of computing a map of the chemical universe where similar chemicals are close to each other and dissimilar ones are far from each other. And then we added where we had 74 different properties, which we had for 600,000 of the chemicals. And this was published. I don't need to go into much detail. In 2018, we were able to predict 9 of the most common toxicity tests, the classical 6-pack plus ecotoxicological test and inhalation toxicology. We've been able to predict 190,000 cases of a chemical where we had a classification and were 87% correct. Now, this was quite a hit at the time. And this was possible because we were not just looking for, let's say, eye irritation data when we predict eye irritation, but we were using one of the key principles of artificial intelligence, which is data fusion or transfer learning. So, because if you know that a similar chemical is a skin corrosive, it is certainly also an eye irritant, even if you don't have the data. And in this way, the different properties inform each other. The software is available through Underwriters Laboratories, a safety standard organization in the US. A year ago, the version 2.0 was released. And interestingly, this was accepted last year by Australia as for their chemical legislation. So you can now, for 5 human hazards, use this computer program to predict the properties. And it's under evaluation by the FDA. The big vision now is to integrate more data. This was all an integration of animal data and some chemical physical properties, but we would like now to go further and use in vitro data beside the in vivo data and chemical data, predicted data from QSARs and convert them to make then prediction not only of the adverse effects, but off-target effects, biomarkers, interspecies differences, and adverse pathways in the end. And this vision we are pursuing now with a new project. We were excited that the European Commission did choose OnTox, which started on 1st of May only. 18 groups are involved. ToxTrack, Tom Richtefeld's company, CutUS, our center, is involved, and we are heading the AI part of this project, which is at the core of here some systematic toxicities. So I think this is an example of showing you that AI is indeed a different way of evidence integration, where the algorithms help us to learn what is the important information, which one we should take to make our predictions, where it is not done by humans, but within the algorithm. I'd like to close with a quote from Stefan Platz. He's the Senior Vice President of AstraZeneca. And he said, AI will not replace toxicologists, but those who don't use AI will be replaced by those who do. And I couldn't say it better. Thanks a lot.
Fantastic, thank you, Thomas. So just waiting for people to pop some questions in the Q&A. So whilst we're doing that, I think I have a question for you myself. So given, Thomas, that a lot of chemical safety testing and regulatory decisions are predicated on physical tests, right? What do you think the major challenges are for artificial intelligence and its acceptability to regulators?
I mean, you have to first see that AI is very much a black box technology. We have still to do a lot about understandable AI, explainable AI. We have to understand how we can show that a given prediction is really right and justified. Because AI will always give you an answer. If you train an algorithm on weather data and you predict the stock market, you will get predictions. But they will not be good predictions because the answer was not in the data set. So it is really about showing that the data contain the answer, and that it was trained sufficiently to really come up with something which is plausible, validating it with cross-validation or better external data sets, and then still have an expert judgment of before you apply something like this. That's, from my point of view, the biggest challenge we are facing before the regulators will be happy to hand over to the computer to come to conclusions. Yeah,
so because I think there's quite a big difference, isn't there, in terms of how we perceive how we do research when we're like, you know, the classic model of doing systematic reviews. You look at a study and you read how the study was conducted, and that helps you assess risk of bias in that study. And then you do that for 20 odd studies, and you come to your conclusion in your systematic review, right? In that way, we're very focused on the details in these individual studies that we have, and dependent on how well those studies are reported for coming to our conclusions, right? But then, obviously,
this is the funnel that we should create. Yeah. I mean, this is a completely different way of doing it. Yeah. As you just say, from thousands of abstracts, we typically drill it down to some 20, 40 papers which really fulfill all of the criteria. That's what we call a systematic review. If we do a risk assessment, we identify a lead study, and all of the rest of the studies we use for interpretation of this lead study. Yeah. What AI is doing is it takes everything into a big bucket and is looking for what are the pieces which are really giving us the best results in case of a supervised learning approach, for example. Yeah. So it's really different. But I think it is very promising to use this, be it as a complementary tool or something which might be able actually to do better than we have been doing in the past.
I just wonder how we bridge that gap then in the minds of people who are, used to doing systematic reviews, and then we're saying, but if you look at all the data and you put it through these machine learning algorithms, right, then you get better answers. But how is that how do we make that process like visible to people, or a process that we know we can trust?
First of all, by comparing, do we get similar data, and where we are possibly wrong with either method to try to understand why we are wrong. AI is not a savior. Yeah? IBM's Watson, for example, developed proposals for treating cancer patients, which would have killed the patients if physicians had not intervened. Because very often there's a bias in the data sets, and if you don't recognize this, it is very difficult to understand that there's a bias in the results. And And that's why we still need the expert. That's why we need quality controls and validation. But working with big data, you cannot create them in the same way. You cannot start carrying out quality scorings. We cannot do all of the stuff we love to do for our systematic reviews. We compensate for this by quantity and by the interactions between the data, which we also include.
Yeah, so this is going to get us back to Jan's presentation. So in what sense is the quality of the, say, the reporting or the information about the ways in which studies were being done, right? How much of this is captured in the big data systems at the moment? And how much of this information do you actually think we need, right? If that makes sense as a question.
Yeah. I mean, the smaller the data set is, the higher the quality has to be to come to good conclusions. But you also need tons of data to compensate for a lot of bias and other things. And there's probably a sweet spot in the middle, which is using curated data, using automated curation, identifying outliers, and these are tools we still have to develop. Yeah? They're not automatically used, but I think these are the tools which make AI even stronger.
Okay, so in that case, I'm going to ask Jan a question about the COVID-19 pandemic and what that has helped us learn in terms of raising the profile of good reporting, for example.
Yeah, I mean, from the clinical side, I think, you know, we have seen a lot of upfront reporting on protocols for the vaccine development, and also the, you know, the statistical analyses that were going to be carried out. And I think this did allow scrutiny of those methods up front. And it also, I believe, helped accelerate approval because the regulatory bodies had access to that information early. So I think that's a positive. I think in the preclinical space, you know, we have had, for example, NCATS have produced an open data portal around COVID. Which also includes, you know, assays, drug targets, screening data, etc., and also their pharmaceutical collection of, I think it's around 3,000 approved drugs, so people can have a look at activity against the virus. So this has really allowed scientists to sort of formulate and test hypotheses and prioritize research. So I think there have been some positives coming out of this, but there is so much data as well. And to Thomas's point, you know, we need mechanisms to be able to access that easily and in a way that's understandable to everybody as well.
Yeah, so I guess in terms of quantity of data, Katja, this will speak to you with the Tox21 study that you did. So you had all that information. I mean, obviously you sifted through over 10,000 studies initially, screened them down to 700, eventually included, I can't remember how it was, but quite a lot.
42. 42.
Yep, in order to understand how we could have predicted maybe the toxicity of triglycerin earlier, say. Do you have any thoughts on maybe how, if you're going to do it again, how you might do it differently? Maybe if we think about Thomas's work with artificial intelligence, would there be ways of maybe either automating component systematic review process or even thinking about taking a big data approach to this kind of work in future?
Yeah, well, and there are certainly tools now that help you automate this. We actually did this as a brute force at the time, but what I would do is I would use the labeling approach in AI to help with some of the AI tools that we use now. They use this human in the loop approach, and that's one of the comments I wanted to make to Thomas's answer, is that all these approaches need human in the loop at some point. So the programs that make a decision and prediction on inclusion or exclusion based on human reviewers' responses, I think are going to give us the most useful data in the end that comes out at the end. If you think about it as a grinder, where in the end you get a sausage, which is your result. your evidence that you will be basing the risk assessment on, right? So that's one comment. And another one, so I would use more of those tools at the front end. The second one is that this case study actually with our systematic review would be an extremely interesting case of where you do put everything in, right? So we had this ToxCast database with, I think, what is it now, 10,000 chemicals or whatever. We only used information on 2 of them, but there is information on all of the other ones, plus the systematic review data, right? If you put it all in, right, would have the regulators, if the regulators looked at the entirety of the evidence, including their submitted studies, would they have come to a different conclusion, right? So that ToxCast in vitro information is not now part of the regulatory submission, right? So somewhere, and at least one of the pharmaceutical companies that I'm familiar with, they look at 800 endpoints before they do rat studies, okay? 800 mechanistic endpoints. So at some point, somebody had that information, right? It's just because it's not required, it's not part of that overall evaluation. So the AI tools here actually could be quite useful if you do have that information as part of the package.
So Thomas, I mean, the risk of asking one researcher how they do it. Sorry. I was just going to ask you for your reaction, basically.
No, I think there is also a simple limit to what we can read and understand as researchers. You asked for COVID earlier, yeah? There's now 240,000 articles which are available as full text, for example, collected by the Allen Institute in April. Yeah? 240,000 articles. Nobody can mine them. Yeah? The AI is the only way of really extracting information on this. But Katja was very nicely saying the same thing I was saying. You need a human to control what is the output, and to control is it plausible what is being extracted here. COVID-19 was also very teaching. When we had in PubMed in November already 75,000 articles about COVID, there were only 300 using machine learning. I was very disappointed at the beginning. And then in February, I looked again, we had upped this to some 85,000 articles, but there were 2,500 using machine learning. And then it came to me, you first need to have the data which you can analyze. AI cannot work on a few initial data sets. You really need enough to digest in order to make sense with these type of algorithms.
So if we were going to say we were going to reconduct that TOX21 study then, like we could imagine just planning it now in 2 minutes, yeah. How do you think we should go about doing that? So Katja and Thomas.
If you do it AI-wise, you would put in all of the ToxCast data, and you would put in all of the literature you can mine on these things and the database, and you would allow the computer the AI algorithm to find what is really distinguishing these two chemicals. That's your supervised approach. And you will see that there is clustering of certain properties, and the computer takes everything into consideration. You don't need to sort out this one. It does not seem important. This one seems to be biased or whatever. The computer will find whether it's meaningful or not if there's only enough data. So it would be a fascinating experiment to see.
It would be really like putting everything in and seeing what I am a little skeptical about the computer doing it by itself. Again, I'm a proponent of having human in the loop here, but it would be interesting as an experiment to see what the computer would come up with, and having it compared to the human approach that we took.
See, I still think that, I suppose, in a way to make these things transparent, the data that goes in, it still needs to be understandable by a human. We can just have laboratories producing numbers and send those numbers to an AI and then hoping for the best, right? So I guess this again brings us back round to reporting standard things. So Jan, in terms of what we've learned from clinical reporting, is there anything in there that will help us go about improving the reporting of the preclinical data, which is the type that we generally deal with in toxicology, that might help with improving our general understanding of how research is done and our ability to evaluate the reliability of its results.
Yeah, I mean, I think you're right. I still think we need some standards for reporting preclinical data, you know, in a, whether that's SEND or something else. And I think, you know, maybe they're not as advanced yet for preclinical data as they may be for clinical data. But as I think Katya's mentioned in the chat, you know, people aren't even obliged to report outcomes. They have to register protocols, but they're not even obliged to report outcomes. So I think that's a huge piece that needs to be addressed, and we need incentives really for people to do that. And you know, we know from what takes place with clinical trials that there are huge gaps in that process, and they have to, you know, researchers have to have a compelling reason to really report this data. So my take home would be that we need guidelines, we need standards, but we also need incentives and possibly penalties if this data isn't reported. It's not a nice way to go, but I think we need to do something to make people report this data. Otherwise, we're wasting so much money and resource, really.
Exactly. It's very frustrating when you go to clinicaltrials.gov and you're looking for information on a drug, and you see that the trial has been marked as completed 5 years ago, and there's no data reported.
Yeah.
And it's, I don't know what percentage of clinicaltrials.gov is there, but I just recently have done that, and it's more often than not, I would say in 80% of cases where I looked, it was not reported.
Yeah. Agree.
So a slightly different flavour of question I think we can use to wrap everything up from Aaron O'Sullivan just in the chat box here. And he asked, do you expect a chemical universe approach to be applied for chemical occurrence and concentrations in the environment?
Yeah, I mean, it's the question is really to get the information in. We have so many chemicals in the environment. We have seen that there's about 350,000 chemicals which are found in 19 countries, chemical repositories only. We see in environmental samples, there's often several thousands of chemicals without data. It's not easy. We've started applying this, for example, to dust samples, and so to really measure, do a mass analysis, a profiling of things we can identify with mass spec technologies. But this is in the very beginning. Yeah. The interesting thing is that the chemistry is not that different. For example, we thought at the beginning we will not be able to predict drug properties, and sense toxic properties of drugs with industrial chemicals toxicity data. But the chemical space is actually very nicely covered. There's no other chemistry. And I have no reason to assume that the environmental chemicals are really so much different from the industrial chemicals. But this all needs to be proven. Yeah, I'm at the moment doing educated guesses, not more than this. Yeah.
Very good. Thank you. So I think probably many people who are in the audience will have another call in 5 minutes. So what I suggest we do is that we thank our presenters now and we wrap up. So Jan, Katya, Thomas, thank you for your time and your slides. This will be posted on the EBTC YouTube channel in the next few days. I would heartily encourage you all to sign up to our newsletter. If you want to do that, you should email Katya at this address, which I'm going to try and pull up quickly. I would just say thank you again, and I hope you have an enjoyable weekend.
Thank you.
Thanks, Paul.